Welcome to this explainer. Today we're unpacking one of the most incredible scientific detective stories of the 20th century. We're exploring the mind-bending reality that the human body is actually a chimera. Seriously, it's a magnificent patchwork of ancient bacteria. So if you think you know what makes you, well, you, prepare to have your perspective totally shifted. We're going to look at how a radical theory, one that was completely dismissed by the scientific establishment, actually proved that the very engines of our cells were ancient invaders.
Here's the roadmap for the journey we're taking today. First, the aliens inside us. Then, Lynn Margulis' radical idea. Next, how endosymbiosis works, followed by the battle for acceptance, and finally, evolution by cooperation.
Okay, let's dive into this first section. The aliens inside us, the microscopic reality.
90%. Think about that for a second. By cell count, 90% of the cells inside a human being are actually bacteria. Now, while they might only make up a tiny fraction of your physical mass, they literally account for 9 out of every 10 cells inside you. We live in this constant state of symbiosis with trillions of unicellular organisms, right? Some are incredibly beneficial, like the ones breaking down your lunch in your gut, and others, well, they cause issues, like the bacteria forming plaque on your teeth. But it establishes an undeniable reality. You are mostly a collection of strangers.
And that brings us to this incredibly unsettling question. If 90% of you is undeniably bacteria, what about the remaining 10%? I mean, what if the rest of you isn't completely you either? What if even your own so-called human cells, your complex eukaryotic cells, are actually ancient patchworks of foreign organisms? Well, this was the exact question that a brilliant young scientist dared to ask.
Section 2. Lynn Margulis' radical idea.
Our protagonist here is Lynn Margulis. She was an exceptionally precocious, brilliant, and honestly, famously stubborn scientist. I mean, by age 16, she'd already entered the University of Chicago, and by 20, she had wrapped up her studies in zoology and genetics. In the 1960s, she set her sights on challenging the most deeply entrenched evolutionary dogma of her time. But to really understand her theory, we need to ground ourselves in this crucial term early on, symbiogenesis. If we look at the Greek roots, syn means together, bios means life, and genesis means origin. Put it all together and it literally describes the creation of new, complex life through the physical coming together of distinct, previously independent organisms. So it's not just species interacting, it's species physically merging to become something entirely new.
And this brilliantly illustrates the core of her work. While she was studying the internal structures of complex cells, specifically mitochondria, which generate energy, and chloroplasts, which handle photosynthesis in plants, she noticed something absolutely staggering. These tiny cellular engines looked exactly like miniature cells within a cell. They were wrapped in their own membranes, they multiplied by dividing in two, and most shockingly, they possessed their own distinct DNA. Margulis realized she had seen these exact characteristics before in free-living bacteria.
Moving on to Section 3. How endosymbiosis works. Let's walk through the chronology of primary endosymbiosis step by step. Billions of years ago, a large proto-eukaryote, a single-celled organism, engulfed a smaller free-living bacterium. Now, normally this is just lunch, right? The larger cell would just digest the smaller one. But here, the bacterium actually avoided digestion. Instead, they formed this mutually beneficial Trojan horse partnership. Over massive stretches of evolutionary time, the engulfed bacterium lost its autonomy, transferring much of its genetic material to the host, and permanently became a specialized organelle.
So why would the host cell keep the invader alive instead of just eating it? Well, because of the massive evolutionary benefits. Billions of years ago, cyanobacteria started filling the Earth's atmosphere with oxygen. And actually, oxygen was highly toxic to early life. But the engulfed bacteria that eventually became mitochondria could not only survive this toxic oxygen, they could actually harness it to create massive amounts of energy. Similarly, the bacteria that became chloroplasts could literally create sugar from sunlight. By keeping these inner aliens alive, the host cells gained an unbeatable survival advantage. They basically transformed these invaders into the indispensable powerhouses of our modern cells.
Section 4. The battle for acceptance, fighting the dogma.
Revolutionizing biology doesn't happen overnight. Let's move to Zapata and see how this builds historically. The idea actually traces back to 1883, when botanist Andreas Schimper first tentatively suggested chloroplasts looked kind of like symbiotic algae.
But the idea just lay dormant until Margulis resurrected it in the 1960s. Her groundbreaking 1967 paper was famously rejected 15 times before finally being published. 15 times! Biologists thought her idea of wholesale genetic swapping and cellular incorporation was just rank heresy. It wasn't until the 1980s, when modern molecular biology and DNA sequencing matured, that she was ultimately vindicated.
The great debate really hinged heavily on DNA. Inside your cells right now, you have two distinctly different types of DNA. Your primary nuclear DNA is bundled up in linear strands. But the DNA inside your mitochondria, it actually forms a continuous circle. And you know what else has circular DNA? Bacteria. Critics tried to argue that, well, maybe both just evolved from an early circular ancestor along separate paths. But as genetic sequencing advanced, this genetic anomaly forced scientists to finally take a radical origin story seriously.
Now, what's really interesting about this slide is the sheer volume of proof. The overwhelming structural and biochemical evidence acted as the ultimate smoking gun. We aren't just talking about shape here. New mitochondria form exclusively through binary fission, just like bacteria. They share specific transport proteins and unique membrane lipids that are entirely absent in human nuclear genes. Basically, the biochemical fingerprints of bacteria were absolutely everywhere.
Which brings us to Section 5, Evolution by Cooperation, a cooperative network.
With the scientific community finally convinced of the biological reality of endosymbiosis, it triggered a profound philosophical shift. There's this stark contrast between two evolutionary views. The traditional neo-Darwinian perspective heavily emphasizes gradual genetic mutation and pure individual competition. It's often summarized as nature, red in tooth and claw, where only the fittest survive through combat. Symbiogenesis, however, presents a very different mechanism for evolutionary novelty. It demonstrates that some of the most major evolutionary leaps, particularly the origin of complex eukaryotic cells, resulted not from gradual mutations, but from symbiotic mergers. It really shows that joining forces can be an incredible driver of evolution.
So the crucial point is perfectly captured in this powerful quote from Lynn Margulis herself. Life did not take over the globe by combat, but by networking. By redefining our understanding of cell evolution, her legacy reminds us that in a world so often seen as dominated by competition, the most profound advancements in the history of life on Earth actually arose from profound collaboration.
And that leaves us with a lingering, provocative thought to conclude our explainer. If the biology we've looked at today is true, if the very cells making up your heart, your brain, and your muscles are actually a cooperative network of ancient strangers that learned to live together billions of years ago, how does this change the way you see your own existence? You are not just a single individual. You are a walking, breathing, functioning ecosystem of ancient partnerships. Thank you so much for joining me today, and keep exploring.